Hey there, guys, it's Mrs. Hallman. Today we're going to be talking about Mendelian versus non-Mendelian genetics. So for this screencast, you guys should have open from Google Classroom your Day 2 genetics notes titled Non-Mendelian Genetics. As you watch this, you should be filling in notes in a level of detail that will help you. Specifically for today, really focus on the examples of each of the different types of non-Mendelian genetics. And perhaps even if you want to have handwritten notes by your side, you'll be able to do some practice Punnett squares as well. We are going to be skipping over the information that is a part of this section that regards blood type, because tomorrow's class is going to be focusing on that. So we're going to kind of skip over that. You won't have to take notes on that. I won't go over it in the screencast, and we'll be meeting tomorrow to learn about blood type. All right, so let's get started. So first thing I want to go through is how it is that Mendelian genetics is different than non-Mendelian genetics. So let's remember that Mendel was the Austrian monk that first kind of discovered that genetics is not just a blend between a mother and a father, that genetics happens in discrete, tiny packets of information we now call chromosomes. He learned that there were dominant and recessive alleles, and in that he kind of put together this set of rules. And those rules only apply to this section of genetics called Mendelian genetics. Mendelian genetics is very simple. We'll learn that as science advanced more and more, and as we learned about more genes, we discovered that there are some traits, in fact, a lot of traits that don't fit into Mendel's rules. Think of them as kind of like your basic alphabet of genetics. And when you get into really advanced writing, you learn that those rules that you learn in English class, you can break them as long as you understand them. So non-Mendelian genetics is more advanced. It happened after the time of Mendel, and actually we'll learn that more human traits are actually non-Mendelian than they are Mendelian. All right, so let's kind of compare these two categories, and you guys can write down main ideas of Mendelian versus non-Mendelian genetics. So first of all, in Mendelian genetics, each trait, so remember, trait is a characteristic. Each characteristic is controlled by one gene. All right, so it's very simple. One gene, one characteristic. In Mendelian genetics, each trait only had 2 alleles. Remember that vocab term alleles means versions of a trait. So for example, in Mendel's pea plants, the pea plant could only be tall or short, and those traits would be either dominant or recessive. Along with that, Mendel said that every trait had to have a clear dominant and a clear recessive allele. So again, that means that the dominant would mask over and cover up that recessive allele. Remember, the only time that we only had a recessive allele show up is when we had 2 alleles, like little t, or little a, right? In Mendelian genetics, really the problems that we were solving with Punnett squares were simple problems, and they always had simple answers. In our last class, we kind of talked about how really when you think about Punnett squares with Mendelian genetics, there's only a couple different versions of Punnett squares you can do before you start noticing that there's a pattern of how those alleles come out, how those genotypes come out. And that's because Mendelian genetics is very simple compared to non-Mendelian. All right, so let's talk about non-Mendelian. So we're going to kind of contrast each of those statements and talk about non-Mendelian. So Mendelian, each trait only controlled by one gene. In non-Mendelian genetics, we learned that some traits are controlled by more than one gene. All right, we'll go over how height, hair color, and intelligence are examples of those. Some traits have more than 2 alleles. So what this means is that instead of a pea plant having a purple or a white flower, sometimes flowers will have a range of colors. So maybe purple, maybe pink, maybe white. All right, we'll learn about an example involving bunnies with that. Dominant alleles do not always mask over recessive. So here's where it gets tricky. We learned so far that if you have big T, big T is dominant, little t does not show up. But in non-Mendelian genetics, we discovered that actually sometimes the recessive and the dominant alleles will blend together. And we'll learn about how that's called incomplete dominance. In non-Mendelian genetics, instead of having simple problems and simple answers, the problems are going to be a little bit more complicated, but here's what's good about that. They're more realistic. All right, we are complicated as humans, which means that these problems are going to be more life applicable, which is kind of cool. All right, so examples that we'll talk about will be incomplete dominance, codominance, multiple alleles, and polygenic traits, and we'll talk about those. We're basically going to go through 4 different examples of non-Mendelian genetics, and these 4 are the ones you're responsible for. For each of these 4, the term in red is the term I want you to kind of associate in your mind. If we were in class together, I have this fun activity where anytime I say the word incomplete dominance, I don't care if it's in a test or a quiz or the hallway, whenever I say the words incomplete dominance, I allow my students to scream the word blended, because I really want your brain to connect those 2 ideas. All right, and we'll talk about why that word blended is a good word to think about with incomplete dominance. Okay, so let's talk about incomplete dominance. Incomplete dominance, let's think about that word incomplete. Incomplete means halfway, but not the whole way, right? So in dominance, typically an allele will cover over another allele. So if it's incomplete, it means it covers over but only a little bit. So what this means is that actually neither allele is dominant over the other. And what that means is that heterozygous, so let's remember heterozygous is when we have like big T, little t. It's when we have a genotype that has 2 different alleles. So heterozygous individuals have a phenotype that is between the homozygous individuals. Oh my goodness, what does that even mean? So when we look at heterozygous individuals, all right, so let's say big T, little t, it means that their phenotype, remember this is their physical characteristic, their physical trait, is in between the homozygous individuals. So that means it's in between big T, and little t, because those are 2 homozygous individuals. So what that means is that we would have kind of a spectrum, like big T, little t, and each of these would have different characteristics. Now, to keep ourselves straight, we don't use the same letters for incomplete dominance as we do normal Mendelian genetics. We don't use capitals or lowercase because remember, big T and little t, that kind of gives us a clue that this is dominant over little t. And because neither allele is dominant over the other, we actually don't use capital and lower cases. Instead, we typically will use 2 different letters. So maybe if we're talking about color of a flower and we're looking at red and white, we will actually use 2 different letters, both capital, which means our genotypes would be RR, RW, and WW. So this would be red, this would be white, and then remember, they have a phenotype. So heterozygous individuals, this one's heterozygous, will have a phenotype between homozygous individuals. So if we have red and we have white, think about art class, you blend them together, RW would be pink, okay? So that's one of the ways we can think about incomplete dominance. So let's go on here and let's get an example. So snapdragons are a type of flower. They're really, really pretty flowers, but they exhibit this type of inheritance called incomplete dominance. Another example of a flower that has incomplete dominance are called Japanese 4 o'clock flowers, and they're very little cute pink and red and white flowers. But essentially in snapdragons, the flower color can either be red, pink, or white. And the heterozygous condition results in a pink flower. So let's look down here at our little key. If we have a red snapdragon flower color, it's going to give us the RR genotype, meaning both alleles are red, flower is red. If we have a white snapdragon, we're going to have 2 white alleles. Okay, so this one and this one are both homozygous. And remember, the heterozygous is going to be the blend between the 2. So if we have 1 R and 1 W alleles, we're going to end up with a pink snapdragon. So this is our heterozygous individual, and that's going to be the blend between the 2 other alleles, okay? So here we go. Let's talk about doing a cross with this. So here's our snapdragons again, and let's actually do some Punnett square work with this. So here we have our original P generation. You guys remember last class we talked about the P, the F1, and the F2. When we cross the the white with the red, and here instead of using the R and the W, they use big R, little r, but remember, it doesn't really matter. These letters are just substituting as long as we have a key. So I very easily could have made this a big W. It wouldn't have mattered. The math and the science would have worked out exactly the same. Okay, these are just variables we choose. So in this example, we have our white individual, we have our red individual of flowers. And when we blend these two together, think about the Punnett square that would look as a result of that. Big R, little r, we would end up with 100% big R, little r, which is what we see here in our F1 generation. So let's imagine we take two of these flowers, we blend them together. This would be the offspring of those flowers. Why does that make sense? Well, let's do the work up here. Let's draw the Punnett square for it. If we had a big R, little r, we would do the Punnett square and it would look like this, which means one individual would be completely white. That's this guy. Two individuals would be heterozygous. Those are those two pink individuals, and this individual would be red. Okay. Lastly, in this diagram, they can say that if you were to take one of these big R, little r's, these heterozygous individuals from our F1 generation and blend it, and breed it with a white flower, so a homozygous white flower, we would end up with a Punnett square that looked like this. So big R, little r, little r, little r. We would end up with 50% of our flowers to be pink and 50% of our flowers to be white. Okay, so that is how incomplete dominant crosses work. We can see that this is an example of us listing that cross between our two F1 generation crosses. So our big R, little r, two heterozygous individuals, we can see the phenotypic and the genotypic ratio in incomplete dominant cases end up being the same. Remember us talking about how we have to list the phenotypes and list the genotypes. Well, in this case, the red, white, and pink are going to correspond with the genotype. So incomplete dominance is one of the only times that that actually works out, that our phenotypes and our genotypes are going to be the same. All right, so let's talk about human traits, because snapdragons are fine, but we want to know about ourselves. We're interested about human genetics. So other traits that we have that are expressed through incomplete dominance is hair texture. So the way that we kind of describe hair texture is that there really are two different kind of alleles for hair texture. There is what we call smooth and there is curly. So if you have smooth, smooth or straight, straight versus curly, curly, this is going to be straight hair. This is going to be curly hair. And SC would be wavy hair. All right, so they blend together. The straight is not dominant, the curly is not dominant. Remember that word blend is what we always think of when we think of incomplete dominance. Another great example of this is skin tone. All right, so if we have somebody that has dark skin tone versus light skin tone, this is an example of incomplete dominance. It's also something that is an example called a polygenic trait. We'll get to what that means in a second. But basically what that means is if we have two medium colored individuals, so this one's kind of a medium colored individual, if they end up having offspring, there is a chance that their offspring would be less dark in pigmentation or more dark in pigmentation, but the greatest probability would be that they are the same color, that they are that same medium pigmentation. So we see that a lot of times with mixed race couples that have offspring with different pigmented skin. A great example of this, and I won't spend a whole lot of time on this, the article is linked for you, is that in 2006, there were actually twin boys that were born, and because both of their parents were mixed, one ended up being very fair skinned, one ended up being darker pigmented. So even though they were twins, they were fraternal twins, and it taught us, and it reconfirmed that skin color is incomplete dominance and that we have a probability of having offspring with different skin color than our own if we ourselves carry those alleles. So kind of cool example. So that moves us to number 2. So our second type of non-Mendelian genetics, and this is going to be called codominance. Codominance. Now the term I want you to always think of, the term that if we were in class you would be allowed to scream at me, is the word pattern. Okay, pattern. Now let's break down this word codominance to give you a little hint. This word co means something. So I want you to think of the word co-captain. If you are a co-captain with another person, all right, so if there are two co-captains, who has more power? Your answer should be neither of them, right? They share. But do both of them have power over their team? Not power, but leadership over their team? Absolutely. If you are co-captain, it means both of you have leadership, but an equal amount, meaning you don't blend the leadership together. It's not incomplete dominance, but you both express, you both have power or leadership over that team, all right? Another way to think of this is if you are a co-worker or co-owner. It means that you're not going to blend your two ownerships together. You're not going to blend your two work together. You're each going to work, but each works the same amount. So I want you to think of the word pattern. That will make sense in a little bit. In codominance, both alleles are expressed equally. So think about that. In incomplete dominance, we said they blend together, which means neither of them really expresses themselves fully. In codominance, both of them express themselves fully. Neither allele is recessive, but instead they are mixed, meaning they are located in different areas. They display themselves in different areas. So I want to introduce you to two different types of animal colorations. The first one is called an erminet chicken. And if I were to ask you a question, say, hey, does this chicken have black feathers? You could say yes. And if I said, hey, does this chicken have white feathers? You could say yes, because the white and black feathers establish themselves in a pattern. We can't say that the black and the white blend together. If the black and the white blended together, we wouldn't have a striped erminet chicken, we would have a gray chicken. So this is how codominance is different than incomplete dominance. Okay, so the traits are shown in a pattern. Another good example of this is the term roan. All right, if you're around horses or if you've ever had an experience with farm animals, you'll notice that sometimes the coloration of horses and cows are quite tricky. And this cow down here, if I said, hey, does this cow have white fur? You could say, yeah, it definitely has white fur. Does it have red fur? For sure, it has like that reddish chestnut brown fur. But if I said, is it mixed together to give us like a light brownish, reddish, pinkish fur? You'd say no, because they express white and red fur. It's just that they express it in a pattern. So you can see that this cow has clear spots, has clear patches where it has red versus white fur. In this horse, this is also a roan horse. And you might say, well, that does look like a blend, doesn't it? It kind of looks like the white and the reddish color blended together. But here's the interesting thing. If you would actually look at the individual hairs on this horse, the hairs are either pure white or pure reddish brown. It's just that it goes red hair, white hair, red hair, white hair, red hair, white hair, so that when you back up, it kind of looks like it's blended together, but it's not. If you were to brush this horse and look at the hair that gets stuck on the brush, there would be red hairs and white hairs, meaning both alleles are expressed equally. They just show up in a pattern, okay? So let's talk about how we actually show the genetics of this, okay? So let's talk about codominance in an erminet chicken. So here we have a chicken that has all black furs, a chicken that has, not furs, what did I say first? All black feathers, that makes sense, and a chicken that has all white feathers. When we breed those two chickens together, the genes are not going to blend, they're going to become a pattern. So they're going to create this erminet chicken. Okay, now in codominance, you will start to see us using different types of letters to represent our traits. Look at my key up here. Now, what I'm going to do is I'm going to use the letter B to represent black, and I'm going to use the letter B with a little 1, and all that means is B prime, all right? Now, let's take a moment just to think about this. If I make a key, it does not matter what bit, what letters, or what symbols I use. Okay, so think about this. I could use B and I could use W. And as long as I knew what type of inheritance I had, and as long as I had this key, it doesn't matter. And in fact, in the next couple of days, you guys are going to do genetics problems where I don't give you the letters. You have to pick your own letters. And as long as you have a key and you follow the type of inheritance, you will be okay. In fact, I always challenge my students, make a weird key. Like for example, what if instead of using a letter, I say black feathers is going to be represented by a circle and white feathers is going to be represented by a square. Couldn't we do a Punnett square using that? So maybe on this side of the Punnett square, we have circle, circle, and square, square. And then as we Punnett, we'd end up with circle. It looks so weird, right? Circle, square, circle, square, circle, square, circle, square. So we would still say this is a heterozygous individual and that these are homozygous individuals. It does not matter what symbol you use. okay? I wanted to introduce you to this idea of prime because that's one of the ways we can differentiate our two alleles. So in our P generation, let's say we cross a black chicken, which is going to be big B, with a white chicken, which is going to be B prime, B prime. We would end up with 100% erminette chickens, all right? 100% erminette. The genotypes would be the exact same thing as the phenotypes. Okay, so that is codominance. All right, the third type of non-Mendelian genetics we'll talk about is called multiple alleles. And for this one, the word that I want, or I should say the words that I want you to remember is actually dumb bunnies, okay? Now, you need to understand something. I like animals. I'm a bio teacher, which means, you know, I love living things. I love plants. I love animals. There are very few animals that I don't like. But one of them is bunnies. I think bunnies are stupid. I think they're mean. I think they're worthless. I think they serve no purpose. And I'm okay with them all dying. I think bunnies are dumb. And if you disagree with me, you're wrong. So when we think of multiple alleles, I want you to think of the word dumb bunnies. And the reason why that's going to help you learn this is that a great example of multiple alleles is how bunny or rabbit coloration goes. So for multiple alleles, before we get to the reason why bunnies is a good example, for multiple alleles, so far every trait has only had two variations. So eye color, blue or brown. Snapdragon color, red or white. Erminette chickens, black or white feathers. So far, every trait has only had two variations. But what we found is that Mendel's idea that every trait only has two versions is not true, that a lot of different traits have multiple alleles. They have more than one. So a great example of that is blood types, all right? And one thing that we need to remember is that individuals will still only individually have two alleles. It's just that they're going to have more options for those two alleles, and that'll make more sense in a second. So the example we're going to talk about is the coat color in bunnies, okay? So I want to start with this picture. All right, these are the dumb bunnies. Like, look at them. Look at their beady little stupid eyes and their short little dopey ears and their big old feet and those razor sharp teeth and claws. All right, and this may or may not, this hatred that I have for bunnies may or may not, may or may not have come from the fact that when I was little, I went to a petting zoo and I held a bunny and I was so excited because I love animals. And I was petting the bunny and out of freaking nowhere, the bunny claws at my arm and zooms across the fairground and we had to go chasing it down and my arm is like bleeding from the stupid razor sharp claws because bunnies are dumb. Why did the bunny run away? I don't know. I wasn't trying to hurt it. I was just petting it. And from that moment on, I decided bunnies are dumb. Don't get a bunny. It's not worth it. In fact, if you see one outside, kick it, right? Anyway, so these bunnies have different coat colorations. You can see that there are four main different coat colorations. All right, the first coat coloration is called a dark gray. All right, a dark gray means that they're going to have this dark kind of blackish brown coloration. Chinchilla, which is kind of a fun one to say because it's a separate animal, is when the coat color kind of is this sandy brownish white. Chinchilla is the average coat color. It's the wild type coat color. If you see rabbits in the wild, like when you see the stupid things like bouncing around your yard and eating your stupid garden, they eat all of my plants. Anyway, that's the chinchilla coloration. Himalayan is when the coat color actually will be darker in areas that are further away from the body. So basically what that means is the tips of the ears, the tip of the nose, and the tips of their stupid little feet are going to be black versus their body color is going to be like a really light white color. And then of course, the all white bunnies that have no pigmentation are going to be albino. Okay, so those are four different coat colors, but let's talk about what those coat colors actually do. Oops. All right, or the alleles for those coat colors. Okay, so let's talk about terminology and how we actually write this. We call that nomenclature. So instead of there being two alleles, so like R and W or black and white, instead of there only being two options for alleles for us to choose. Now, if you look, there's four. And basically, our genotypes always contain 1, 2 alleles. So instead of only having 2 alleles to choose from and only having 3 combinations of genotypes like RR, RW, WW, we now have more combinations of genotypes. So big C, this is a capital C. So it's really important that as you write this, you write big C's different than your little c. So I'd suggest still writing lowercase letters in cursive. All right, so big C is going to be dominant. This is the most dominant. Then we have little c. So I'm going to draw this as a cursive little c. We have little c with a superscript CH. That's going to represent chinchilla. It is recessive to C. All right, it's recessive to C, but it's more dominant than the alleles underneath it. So next we have little CH. This stands for Himalayan. That's the dark nose, dark ears, dark feet. This is recessive to both big C, that full color, and CCH, which is that chinchilla color. But it's more dominant than the one underneath it, which is just little c all by itself. And little c all by itself is going to be albino with no color. So it is recessive to all other alleles. All right. So let's talk about how we actually get our genotypes. Again, remember that genotypes always have 2 alleles, but now we can choose from any 4 of these alleles to make our genotypes. So for example, let's start going through the many, many combinations. We could have big C, big C. We could have big C, CCH. We could have big C, CH. We could have big C, little c. All right, let's keep going through combinations. We could have little CH, little CH. We could have little CH, little CH. We could have little CCH. We could have little c. All right. If we keep going down, we could have little CH, little CH. We could have little c, H, little c. And we could have little c, little c. So these are all of the genotype combinations we could have. Now, what are the phenotypes? So these are genotypes. Remember, those are the genes that those rabbits carry. What are the phenotypes? So that's how that trait physically looks, a physical characteristic. Big C are both dominant alleles. So this is going to be full color, so that dark rabbit. Big C, this one is going to cover up. It's going to be more dominant than little CH. So it's going to be full. Anytime there's a big C, so this one, this one, this one, this one, it's going to be dominant. So all of these are going to be full. Those dark bunnies. Now for this one, we have little CH. We have little C, CH, little C, CH. So this is going to be that chinchilla coloration. So that kind of sandy color. Then we have little c, ch, little ch. This is more dominant than this one. So we're going to end up with that chinchilla coloration. Same thing here. It is dominant over albino. For this one, little ch is going to be that Himalayan coloration. And so is this one because it's going to dominate over that albino. And this is the only combination that's going to give us that albino bunny coloration. So these are all of our phenotypes. Now let's look at how this works out when we're actually doing bunny problems. So if we're determining the phenotypes of these bunnies. I don't know why you would waste your time trying to do that, because what a waste of time. But let's just say that we have a, let's say we have a chinchilla that happens to be C, CH, CH. And let's say we cross that with a Himalayan, which happens to be CH, little c. All right. So if we were to do that Punnett square, set it up like normal. So we'll put the little c, ch, little ch, little ch, little c. And we would do the Punnett square. Notice that I always put the more dominant allele towards the front, kind of like we always put the capitals first. Okay, in this particular example, these two individuals, little c, ch, little ch, would be chinchilla. In this, little c, ch, and little c chinchilla. This one, little ch, Himalayan. This one, little c, Himalayan. And you could do all different combinations with this, but that's essentially how multiple alleles works, okay? Now that leads us to our last type of trait we're going to talk about, which is polygenic traits, which means many traits. So the term I want you to remember is just to break down this word. So if we break down the word poly, it means many. If we break down the word genic, it means genes. So this means many genes or many traits, okay? So these are traits that are controlled by more than one gene on more than one chromosome. So a great example of that would be height, weight, intelligence, and personality. These are all our most complicated traits. It's really hard to walk up to somebody and say, hey, you have a dominant personality and your dad had a non-dominant personality, therefore your kid is going to have a dominant personality. There's no such thing as just having one trait that controls your personality. It's many different genes on many different chromosomes that are going to control that. So polygenic traits, we don't typically do Punnett squares for. We don't typically list out alleles because it would be way too complicated. All right. However, what that means is that most human traits are polygenic. We are complicated people, guys, which means that it's really hard for us to look at the traits in our body and to narrow them down into either this or that. Most of us, if we stand in a line, are going to be in between versions of that, which means our traits are controlled by many different genes. So that's why human genetics becomes even more tricky. That wheel of all the traits that you guys learned about the very first day of our lesson of genetics, where you guys figured out like attached versus detached earlobes, hitchhiker's thumb versus non-hitchhiker's thumb, cleft chin versus non-cleft chin, dimples versus non-dimples. Those traits are some of the very few human traits that are actually just one trait. The majority of our other traits are polygenic traits, so they're a lot harder to figure out. Okay, so in summary, the 4 types of non-Mendelian genetics that we learned about today are incomplete dominance, codominance, multiple alleles, and polygenic traits. The last thing I want to go over with you, again, we're going to skip over blood type. That's what we're going to learn about tomorrow. The last thing I want to go over with you is genderized traits. So how is it that our gender affects the traits that we have? So what we're looking at here are 2 karyotypes. What's a karyotype? Well, a karyotype is when we take one cell within our body with all of the chromosomes in there. We smash it on a microscope slide, we stain it, and then using a computer, we line up all the chromosomes with their pair. Remember, from each of our parents, we get 1 chromosome of each of the 23 sets that we have. So this might be from mom, this might be from dad, this might be from mom, this might be from dad. And all the chromosomes are kind of the same length and they have the same stripe or banding pattern as the other. So you can see down here in number 22, they're about the same height. This one's a little curvy. All right, but you can see that they both have a dark spot right here. And that dark spot is their banding pattern. So they will carry actually the same genes. Now what we see is that we have 22 pairs in each of these karyotypes except for this last pair. This last pair are going to be our X and Y chromosomes, and that is how we determine gender at the chromosomal level. All right, if you take anatomy and physiology or AP Bio with me, we get into the nitty-gritty of how gender is actually determined and how gender really is kind of like interesting to study. It's so cool. There's so much cool science that determines it. And we've learned more and more over the past couple decades that genders really can be kind of on a spectrum, which is kind of interesting. So karyotypes from a male versus karyotypes from a female. All right, so when we're looking at this karyotype, we have an X and a Y. Very, very different length chromosomes. So this one's kind of itty bitty, this one's kind of long. The Y chromosome is going to be very, very short. In this one, we can see 2 identical X chromosomes. So the answer to this is that the one on, let me clear this real quick. The one on the left is going to be our male karyotype, meaning that it is going to have an X and a Y. This is the symbol that represents male. And the karyotype on this side, XX, is going to be female. So what does that mean? Well, each individual has 2 sex chromosomes that will determine if they are male or female. All right. Chromosome in that karyotype that is non-sex chromosome. So if I go back a second, we have all of these autosomes. So chromosomes 1 through 22, all of them are going to be autosomes. These are our only sex chromosomes. Okay. So a sex chromosome is going to help us determine gender, and they're either going to be X or Y chromosomes, okay? So an X and Y chromosome both contain genes that are needed to develop into a male or female. Specifically, if you're interested in gender development, the Y chromosome has a tiny little gene on it called the SRY gene. And that SRY gene is actually one of the genes that will be expressed really early on in fetal development that will either determine your sex genitalia, your gonads, to be testes or ovaries. So that SRY gene will start to program that developing fetus as either a male or female. However, the X chromosome has a lot of other genes on it, so I'm drawing some potential genes that may have nothing to do with gender. And we're going to learn about some characteristics that live on the X chromosome, which means if you are females, you have 1, 2 copies of those X chromosomes. If you are male, you only have 1. So we're going to talk about some of those examples and how they relate to you. One of the best and favorite examples is male pattern baldness. And I know that a lot of you guys have heard that if you want to know if you are male, whether or not you're going to go bald, you can't look at your dad. You have to look at your mother's father, which is kind of interesting. Also know that male pattern baldness is only expressed if you are male. So females, thank the Lord, we don't have to worry about thinning on top. All right. Now, that's not to say that females can't go bald. There are other conditions, some genetic, some not, that do result in females losing hair density. There's several different examples of those, but we're not talking about those today. We're just talking about the classic male pattern baldness where you lose hair from the very top of your head and you end up with the ring or the crown around. This is my personal tactic or my personal job, I feel like, as a teacher, is just to teach you guys never, ever, ever do this. So gentlemen that are watching the screencast now, it is never acceptable for you to have a comb over that looks like this. If ever you are in a situation where you have male pattern baldness, you remember your bio teacher in love saying, don't try to comb it over, just shave it off, right? It looks so much cooler when you just shave it off. So let's talk about these sex-linked traits. Females, because we are XX, have the ability to be heterozygous because we have 2 copies of the X chromosomes. So here's our 1, 2 X chromosomes. And on each X chromosome, we're going to carry a allele. So we could carry big B, big B. We could be homozygous. We could carry big B, little b. We could be heterozygous. Or we could carry little b, little b. We could be homozygous again. So let's just say that this individual is going to be big B, little b. All right. The way we write that is we actually write the X chromosome and we put the allele as a superscript. So we write X with a big B on top of it and X with a little b on top of it. That's representing this chromosome, that's representing this chromosome, and here's our genotype. Now, that's all well and good. That's pretty close to what we've been doing so far, but now we run into the issue with males, don't we always? Just kidding. All right, in males, they only have 1 X chromosome, which means they can only have either a big B or a little b. They do not have the opportunity to be heterozygous. So what does that mean? Well, it means anything that they have on their X chromosome, they are going to express. They don't have the option of being X big B, X little b, and having this big B cover up the little b. All right, whatever the males get on that X chromosome is what they are going to express. So why does that matter? Well, when we talk about inheritance, that's going to affect some of those X-linked chromosome or X-linked traits. So if we remember, let's do some review questions. How many sex chromosomes does each human have? Think about it. Good. So the answer to that would be 2. They either have XX or XY. Which sex chromosomes do females have? Think about it. Good. XX. Which sex chromosomes do males have? Good. XY. And what is the term for a picture of all of the chromosomes in a person's cell? It's going to be a karyotype. All right, so let's talk about sex-linked traits in this example. So I'm going to write it down on this Punnett square because it's our first one together. My key here is that my X big B is going to be normal hair. My X little b is going to be bald. All right, so let's say we cross 2 individuals. Let's talk about what this individual is. First of all, gender, male or female. If it's XX, it's going to be a female, right? So female. This one, XY, going to be a male. All right, now this individual is heterozygous. She carries a dominant and a recessive B. He isn't heterozygous or homozygous because he only carries one allele, which means, unfortunately, this homeboy is going to be bald. Sorry about your luck, buddy. All right, so what we're going to do is we're going to set up this Punnett square just like we do any other Punnett square. We're going to put each allele in a different box going around the Punnett square. Over here is going to be my male. All right, does it matter which side I put each on? Not at all, not even a little bit. All right, and then as we Punnett, here's what's going to happen, and this is fascinating. Okay, so here we end up with an individual. Is this going to be male or female? It's going to be female, right? XX, but she is going to be heterozygous. Okay, next one, X big B, or sorry, X little b, X little b. So she's going to be homozygous for baldness. Now listen, is she going to express male pattern baldness? No, because she's a female. So we have other traits that cover up that. So she's not going to be bald herself, but what it does mean is that her sons are going to be bald. All right, next one, we have X big B Y. Now, first of all, this individual is going to be a male, and he is going to have a full head of luscious hair because he is a big B. This individual, however, is going to get that recessive little b, and this one is going to be a male who is bald. Okay, let's talk about this female. If she is X little b, X little b, let's say she has offspring with a male who is bald. All right, this is a classic example of where guys freak out because they're like, oh, my dad has hair, therefore I'm going to have hair, but they're wrong. So here's our female. We're going to put her up here. X little b, X big B. Here's dad with X big B and Y. So here's the deal. First female is going to be heterozygous. Second female is going to be heterozygous. Here's where things get tricky, and here's where it's a really important concept. In this individual here, this male, who does he actually get his X chromosome from? From mom or from dad? The answer is from mom, right? Every male that walks the surface of the earth got his chromosome, his X chromosome, from his mother, which means every male is going to have more traits related to his mother, all right? And certain sex-linked traits he will always get from his mother. So this individual, bald. This individual, bald, all right? So this is a classic example of where the female actually gave the traits to her sons and the male would not experience that trait himself, okay? So that would be male pattern baldness. That's an example of a sex-linked trait, okay? So I put the actual Punnett square here, so when you click through to review, you have those answers, okay? Another example of sex-linked traits, we're not going to do an example of this because we're running out of time. Another example would be hemophilia. This is a clotting disease where normally if we get a paper cut or if we have a bruise, we'll start to lose blood, but then our blood clots right away. Somebody with hemophilia lacks that clotting factor. So unfortunately, what that means is that as soon as they have a cut or a bruise, they don't stop bleeding, which is really dangerous. And this is a sex-linked trait. All right, so here we have an example of a female that is heterozygous for hemophilia and a male that is dominant for hemophilia. Now, hemophilia can affect males and females the same way. So it's not like male pattern baldness that only males are going to express it. Both males and females can. So let's put this cross into the Punnett square, and we can see that our individuals are going to come out that both females are going to be normal. They're going to be able to clot blood normally, but that's because one of them is homozygous and one of them is heterozygous. So this dominant allele, this X big H, is going to essentially save that female. That's going to be the dominant that's going to give her that normal blood type. The males are going to be 50-50 hemophiliacs, right? So this guy is going to have hemophilia, this one is not. So what does that all mean? Well, that means that in males, sex-linked traits are more common. So sons get sex-linked traits from who? From their mom. And is it more common to have males with hemophilia or females? Definitely males, because they only have that one X chromosome. Another example of sex-linked traits is color blindness. And have you guys ever realized that when you look at the population who has color blindness, males have it more often than females? And the reason for that is that it is a sex-linked trait. It's on that X chromosome. So this is a common eye test for color blindness. The most common other color blindness is red-green color blindness. So if you look at this image to the right, you should be able to see a number. And if you don't see a number, don't freak out. It could be that the screen coloration on your Chromebook is not working. But if you go to a doctor and they give you this official test and you can't see the number 5, you might have red-green color blindness. These are other tests. So on the top left, you should see 25, bottom center, you should see 56, and the top right, you should see 45. So these are tests for deuteranopes who are not able to differentiate between red-green color blindness. And you should see the number 6 and then the number 8 on these. All right. So we are going to stop here for the day. This was our lecture on non-Mendelian genetics. We've covered the 4 different types of non-Mendelian genetics: incomplete dominance, codominance, polygenic traits, and multiple alleles. And then we covered sex-linked traits. So I hope that was helpful, and I'll see you in class.